Yeah, now I want to show you some examples for the use of mark correlation functions based on José's data. And first I show you the data you need. So you go to the folder where the data are placed and where the Programita version is placed. And in this folder you have 4 different data sets. And here in this case the question is that we have droppings of animals which have different seeds in it. And in this type of analysis we want to know if there's a spatial correlation between the number of seeds of one species relative to the number of seeds of another species. So here we have a number of data sets which have basically the data for pairs of species. Yeah, so for example, we can look at this data set here, Todos, Pera, Palma. And if we go to this data set here, if you look at this, then you see again in the first line, you see the dimensions of the plot and the number of data points. And then you have in the first column again the x coordinate, the second one is the y coordinate. Then this is slightly different. Then you have here a number of 1 and 2. This means this is a point where you can have seeds of species 1 and you can have seeds of species 2. So this is needed so that Programita knows this is a type of a bivariate analysis. And then in the fifth column, then you have the number of seeds in this particular dropping. So you see the first droppings, they all have 0 seeds of species 1 and of species 2. But then you see this dropping at this particular coordinate has 83 seeds of the first species. And this one here has only 4. And then there's one which has 150. So it means there's quite a variability in the number of seeds in the droppings. So lots of them have zeros and others have quite high numbers. And then if you go down, you see even cases where you have seeds of both species in. Yeah, or here in this case 22. Yeah, but now the question is there any spatial relationship between the seeds of the two species inside the spatial pattern of the droppings? Yeah, and to answer this question we use the mark correlation functions. Yeah, and to do the analysis you click of course again to Programita, then you see Programita here. And now you have a mark correlation analysis, means you have to go here to mark correlation functions. And now you see here all the different data sets and we go for this Pera Palma one. You click here, then you have to click here. This is data list with coordinates. Then you get again the specifications of the pattern and the bin width. We can do as in the random labeling analysis here with 3 and we have a ring width of 5. Let's see if this is a good setting. Yeah, and then you see now the mark correlation function appearing here. And here of course now you see the pattern. And you see there's quite some heterogeneity in the distribution of the number of seeds over all the droppings. Yeah, in this analysis we selected the mark correlation function. This is basically the product of the number of seeds of the first species in the first point with the number of seeds of the second species in the second point, which is distance r apart from the first point. Yeah, and then this mark correlation function would have value of 1 if they are independent, and if they have an aggregation or attraction, then they have very large positive values, as you see here. But we don't know if they are statistically significant. For this reason, we now go again calculate simulation envelopes to select the random marking null model. And here in this null models we have several possibilities. The thing is, because the droppings of the animals contain the seeds already. So it wouldn't make sense to separate the seeds and distribute them randomly over different droppings. So what we do here, we need a null model so that we can, say, randomize the marks, so the number of seeds, that we randomize them together. So that means each package of seeds of species 1 together with seeds of species 2, they are kept together but then are randomized over the points or shuffled over the points. So this is this very specific null model we use here. Yeah, and then we run the null model. And then you see here the simulation envelopes, which look quite wild if one uses only 39 simulations. So we do a bit more. 199 with the 5 lowest and highest. This goes really quick. Yeah, and then you see still quite stochastic simulation envelopes, but you see there's a really clear effect of aggregation at small scales up to distance of about 20 meters. So that means the seeds of these two species are usually distributed in the same areas or in the same spots. So there is some type of correlation between them. To see this a bit clearly, more clear, we go to this goodness of fit test icon. Then we look at the univariate analysis with the student transformation. Yeah, then we see now quite nicely that there is a significant effect at small distances. Yeah, then of course you could also use other mark correlation functions. There's, for example, the R mark correlation function. And this one is very similar to the first one, but evaluates only the number of seeds of the first species, but under the condition that there's a dropping a certain distance away. And here you see exactly the same effect. So there's the type of spatial correlation between the seeds of these two species. Yeah, and then if you like, you can also do the analysis with another data set, maybe this Palma lentis, the first one. Then you run the whole thing. And then you go to the mark correlation function. Yeah, and then you see this type of strange pattern. You have a peak here about 40 meter, spatial association of 40 meter. And this is probably just these two clusters of droppings. This could be from the budger. This could be the latrines of the budger, which have lots of seeds in. So this creates this strange positive association. Yeah, and then you can play around with the data sets and compare it with our paper. And if you get the right settings, the ones which I selected, hopefully, so figure 4a, for example, the settings, we use a min width of 10 meter. So then let's see how it looks. Yeah, it looks not too much different. There's a very strong aggregation here at small distances. Yeah, and this is exactly the graph as it appears in the paper. Yeah, so I hope you have lots of fun to try Programita to redo this analysis and to learn how to use mark correlation functions.